Hello, everyone. Welcome to today's live broadcast, Hallmarks of Cancer: Detect and Quantify Cell Death Signatures with High Content Imaging. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Molecular Devices. In today's webinar, presenters will discuss two diverse approaches for using automated microscopy and image analysis to measure different and specific indicators of cellular stress leading to possible cell death. Dr. Oliver Kepp from Institut Gustave Roussy will share his strategy of immunogenic cell death fingerprinting for the purpose of identifying novel anti-cancer agents. Oliver Kepp received his PhD in 2006 from the Humboldt University of Berlin and the Max Planck Institute for Infection Biology in Berlin, Germany. He then joined the lab of Guido Kramer in Paris, France, as an EMBO postdoc fellow. In 2012, he became an INSERM staff scientist, CR1 level. Currently, he is co-directing the systems biology platform at the Gustave Roussy Comprehensive Cancer Center, where he investigates several aspects of immunogenic cell death, focusing on systems biology approaches. For the second portion of the webinar, Jane Hesley, application scientist for Molecular Devices, LLC, will illustrate high-resolution and high magnification automated microscopy coupled with tailored image analysis for overcoming the challenges associated for segmenting cells of complex shapes such as neurons. Jane Hesley is an applications scientist for cellular imaging at Molecular Devices, LLC. She has more than 6 years' experience developing cell-based applications using Image Express high-content imaging systems and Meta Express analysis software. Most recently, her focus has been on utilizing stem cell-derived hepatocytes, cardiomyocytes, and neurons for toxicity studies of cell viability, apoptosis, morphological changes, mitochondrial damage, and autophagy. She has a BS in animal science and an MS in biology. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce our first speaker, Dr. Oliver Kepp.
Thanks, Judy. Thanks for the nice introduction. First of all, I would like to thank the organizers, especially Molecular Devices, for giving me the chance to present our recent data on immunogenic cell death fingerprinting. And I would like to present one successful screening approach that we did lately, which was mainly conducted by Laurie Mangère and Erika Vakeli in the lab, and in which we used HCS automated microscopy to analyze the hallmarks of immunogenic cell death. So immunogenic cell death, it is probably best explained on the anti-cancer vaccination model that we used in the lab before we used microscopy to analyze a certain amount of anti-cancer agents. So what we are doing is that we're looking for a bystander effect that chemotherapy can induce while specifically activating the immune system of the host or in clinical conditions of the patient. So the anti-cancer vaccination model that we used consists of an in vitro treatment of cells that we then use to vaccinate syngeneic mice. Upon 1 week of letting the immune system going for a response, we re-inject cells, we re-challenge the mice with living cells of the same kind, and then we measure the absence of tumor growth as an indication for an anti-cancer vaccination efficacy. So this we have been doing quite widely. We used different model systems, and we published this during the last, well, let's say decade in quite a number of high-ranking papers. In these papers, we actually identified parts of the pathway that is needed to mount an active anti-cancer immune response. So these parts of the pathway that are involved include T cells and dendritic cells, certain cytokines and their specific receptors, as well as danger receptors of the P2Rx7 and TLR4 type. Because the effect, the anti-cancer vaccination effect that we see when using in vitro killed cells is entirely absent in mice that lack parts of this pathway. This effect is also absent if the tumor cells that we use for the vaccination lack either the possibility to expose correticulin on their surface, or lack the ability to release ATP upon an induction of autophagy. So all these parts of the puzzle are nicely depicted here in this scheme that we lately published in the Annual Reviews of Immunology, in which again we have the therapy-induced stress, so the chemotherapy killing parts of the tumor. These cells undergo a heavy ER stress, they undergo autophagy, and in the end they will suffer and die. These three signals together lead to the exposure and release of coreticulin on the surface of the dying cells, to the active autophagy-dependent secretion of ATP, and to the release of HMGB1 at late stages of apoptosis when the cellular membranes are disintegrated. These signals are now perceived by the dendritic cells of the immune system by specific receptors. Coreticulin binds to most likely CD91 on dendritic cells, ATP ligates purinergic receptors of the P2Rx7 type, and HMGB1 binds to TLR4 on dendritic cells. All of these signals together are necessary to have the antigen transferred from the dying tumor cells into cells from the immune system, into dendritic cells. These signals are also necessary to confer a proper antigen handling within the dendritic cells. Once the dendritic cells are moved from an immature to a mature state, they release IL-1 beta, which in turn leads to a polarization of T cells, an activation of cytotoxic T lymphocytes, majorly of the CD8 plus type, and to release of interferon gamma, which in the end leads to tumor lysis. This tumor lysis then, as it is a specific anti-cancer immune response, helps to destroy dormant tumor cells or tumor stem cells that have slipped the initial therapeutic approach. As this anti-cancer vaccination model, which I presented in the first slide, was very time-consuming and also very expensive, and it allowed us only to screen a certain number of anti-cancer agents, we switched our strategy towards a fluorescent biosensor detection, which allowed us to enormously scale up the throughput in our system, and which allowed us to screen compound libraries to detect hallmarks of immunogenic cell death. So what we have been doing is that we constructed cells that do express coreticulin GFP together with an H2B RFP, which makes image segmentation very easy, because the H2B RFP always stays in the nucleus, whereas the coreticulin GFP changes its location. So the coreticulin moves from its endoplasmic reticulum location towards the surface of the cells. So what we're looking at is the translocation of chirality from the ER towards the surface of the cells. We also use a chemical stain, which is called quinacrine. With quinacrine, we are able to mark ATP-containing vesicles inside the cell. And this ATP-containing vesicles, the number of vesicles and the intensity of the quinacrine staining massively decreases when ATP is released from the cells in the conditions of immunogenic cell death. We also constructed a biosensor cell line that co-expresses HMGB1 with H2B-RFP. So the HMGB1, which is a non-histone binding protein in the nucleus of the cells, can be easily detected, and its release can be followed by image analysis. All of our biosensor cell lines, this is some of the ones that we have in the lab, which we use for the ICD screening, but we have a whole battery of biosensor cell lines that we can use to detect cellular death and cellular stress conditions. All of these biosensor cell lines have been constructed in a lentiviral-based fashion, which allows to easily customize different cellular models and to go from wild-type cells to a biosensor cell line in a shortness of time. So what we have been using in this case here, this is an in vitro gene-based system which we use to clone our gene of interest coupled to a fluorescent protein. We produced lentiviral particles and then we transduced our cells. This coupled to single cell cloning and growing back the colonies allowed us to easily generate fluorescent biosensor cell lines. One example which I would like to show you here is the fluorescent biosensor cell line that we generated for the detection of HMGB1 release. So as you can see here, what we did is that we used HMGB1 GFP expressing cells and we followed those over time here in this sequences from a movie. And what you can see is that the HMGB1, upon treatment with mitoxantron, which is one of the prototype immunogenic cell death inducers that we use in the lab, the HMGB1 moves from its nuclear location. It's a two-step process, so it moves from its nuclear location first to the cytoplasm of the cells, and then later on it's completely released into the surrounding of the cells. So using this cell line co-expressing H2B-RFP, which marks the nucleus in red, we were able to do image analysis, and we converted this effect that we see in the that we saw in the movies into numbers, which is necessary to conduct a screening approach, of course. So as you can see, the control cells do co-express HMGB1 and H2B-RFP, whereas the mitoxantron treated cells do show a release of HMGB1 GFP. So basically, you go from a green and red expressing cell to a cell that only retains the H2B-RFP in the nucleus, whereas the GFP is released to the surrounding and therefore becomes invisible in the image analysis. And this is an effect that you can easily quantify and put into numbers and statistics. So what we did is that we applied this approach looking for chirality GFP, for the release of ATP, and for the release of HMGB1. And we validated our approach by using different methods to detect the same phenomenon. So we had a look at the effect that chirality in control cells is expressed in the endoplasmic reticulum, and then upon treatment with mitoxantron moves to vesicular-like structures very close to the membrane. We saw the same effect with a construct that expresses ERP57 GFP. ERP57 is another ER protein that co-translocates with charaticline to the surface of the cells during immunogenic cell death events. And we also used ordinary antibody staining in FACS assays in non-permeabilized cells to show that this effect can be seen in different methods, so in FACS and in image analysis. We also correlated the effect of induction of autophagy, which you can see here in this GFP LC3 expressing cell line. We correlated this with the release of ATP, here visualized by the chemical staining with quinacridine, and we also used another biosensor cell line that we got from a collaboration, which is a thread-based detection system which detects the concentration of ATP within the cells. If ATP concentration is decreased, the construct undergoes a conformational change and the thread is lost. We then correlated the release of HMGB1 with the uptake of an inclusion dye. So we correlated HMGB1 release with the permeabilization of the plasma membrane. And as you can see, most of this morphological phenotypes nicely correlated with each other when looking at the very small library that we screened for this purpose. So the drug screening pipeline that we are using in the lab and that we also have used for the project that I'm going to show you in a minute is the following. We generated our reporter assays in the project for the ICD drugs. This was the charaticline, HMG1 and ATP biosensors. We then screen compound libraries that some of them we commercially bought and others we got from collaborations with the NCI or our industrial collaborators and partners. We are then doing HTS imaging, so we're using robotized sample preparation together with the automated imaging. In this case, most of the images have been acquired within Molecular Devices IXM machine. We then do the analysis of all that data that we generated during the screening approach. Therefore, we use the sophisticated statistical software which is called R. And all the primary hits that we get from the initial screen, we validate in counter screens, meaning that we're using assays that are not image-based, but we detect charaticline with an antibody staining and FACS assays. We detect HMGB1 with a commercially available ELISA kit, and we detect the release of ATP with a luciferin-based assay. And from all that compounds that we screened initially, we narrowed them down in the validation screens, and from all that compounds that made it to first and primary screens, we do the in vivo validation, meaning we go back to the initial model that I showed you in the very first slide, and we make sure that the effect we see is really based on an immune system related animal. Because only in a living organism which actually has a immune system up and running, we can detect the real importance of the drug in the context of an immune response. And in the project that I'm going to show you in a minute, we had the benefit, we could benefit from the clinical files that we have here at Gustave Roussy, so we were able to conduct a clinical retrospective study, which we'll see in a minute. So what we did in this project on immunogenic cell death inducers is that we used an FDA-approved chemical drugs library, and we screened for the hallmarks of immunogenic cell death, meaning that we were looking at different time points, at different concentrations for the release and exposure of immunogenic hallmarks. We ranked all this, so we did the Z-scoring of all the hits that we found, and we ranked those in a hierarchical fashion. We did a cumulative Z-score to have a look for all those drugs that have the capacity to bring up all these immunogenic hallmarks together. And when we had a look at the upper 1% percentile of the screens, we found, to our relief, we found quite some compounds from the group of anthracyclines. This is very confirming, because this is something that we initially found in the 2006 Nature Medicine publication in the in vivo model. But apart from those already confirmed hits, we found a group of chemicals that belong to the cardiac glycosides cluster. Those cardiac glycosides were then further validated, so we focused on two compounds from that group, which is digoxin and digitoxin. We focused on those because those have been, and digoxin still is, have been widely used in clinics. So this was the most interesting for us. We validated that the images that we got from the image-based screen really shows the phenotype that we were looking for, and we then went on with the validations, and we used digoxin and digitoxin in a dose-dependent fashion, and we measured the exposure of carradicolin by FACS assays, we measured the release of ATP by luciferase assays, and we measured the release of HMGB1 by commercially available ELISA assays. And as you can see, we had a nice dose-dependent release and exposure of these immunogenic hallmarks when using both prototype cardioglycosides, digoxin and digitoxin. To make sure that this is not a cell type specific effect, we used quite a large number of different cellular models, and we used the prototype cardioglycoside, which is digoxin. This is the one that is still used in clinics, and this is then also the one that we used for our retrospective clinical study. And again, we used a dose-dependent assay, so we used different doses of digoxin, and we validated that digoxin not only in our U2OS, which is human osteosarcoma-based biosensor assay, had an effect, but that it also had an effect on different other cellular models, including cervix carcinoma, colon carcinoma, lung carcinoma, and breast carcinoma. And then what we did is that we combined digoxin with a non-immunogenic cell death inducer, which is mitomycin C in our case, and we validated that the combination of non-immunogenic cell death inducer plus digoxin, which brings the effect of immunogenic cell death, has the ability to cause an anti-cancer vaccination efficacy in our in vivo model. So what we did is that, what I explained in the very first slide, that we killed the cells in vitro. Then we used these in vitro killed cells as a vaccine to vaccinate immunocompetent mice. After 1 week, we re-challenged these mice with living cells of the same kind, which are absolutely potent to form tumors, as you can see here in only PBS control-treated mice. So basically, none of the mice remained tumor-free. All of these mice grew tumors, as you can see here. We pick it down here. Then, when we combined the mitomycin C together with digoxin or together with digitoxin, we saw that up to 80% of the mice were kept from forming tumors, meaning that an immune system mediated effect caused vaccination against the re-challenge with living cells of the same kind. Further went on with our in vivo validations, and we used mice in which we grafted tumors, and then we followed the tumor growth in immunocompetent and in immunodeficient mice. And as you can see here, the co-treatment of mice together with mitomycin C together with digoxin or digitoxin caused a massive and statistically significant decrease in tumor growth. And this, very importantly, this effect is entirely absent when we used immunodeficient mice. So this clearly shows that this system, that this effect is immune system related. Now, as I said, as we're working here in Gustave Roussy, which is the biggest European cancer hospital, we were very lucky that we had access to patient files. And what we are doing in this retrospective clinical study is that we were looking for patients that have received cardioglycosides during the chemotherapeutic approach. And we case matched those with patients that have only received chemotherapy during the same time. Of course, this was the same tumor stage, the same sex, the same age, everything was case matched. And interestingly, what we saw, and initially was also kind of unexpected, when we had to look at the patients that have received cardioglycosides due to an underlying heart insufficiency, those patients that have two potentially deadly illnesses, so heart insufficiency and a cancerous disease, those patients have an increased overall survival as compared to case-matched controls patients. So there was a statistical difference when looking at these two groups of patients. When we did the subcluster analysis, we found that this effect is entirely absent in patients that have been treated with an anthracycline, which is an immunogenic cell death inducer per se. But when looking at patients that have received non-anthracycline-based, so non-immunogenic treatment, in the presence and in the absence of cardioglycosides, we could see a statistically significant increased overall survival of the co-treated patients. So this was very, so we were very happy that we had access to these patient files, because the retrospective clinical study proved that the in vitro image-based approach that we initially have chosen detected a drug that has the capacity to cause immunogenic cell death in vitro, in vivo, and also in patients. So this last scientific slide summarizes the strategy that we have been developing and that we are still following in the lab, which is the necessity for drugs, for successful anti-cancer drugs, to cause ER stress, to cause autophagy, and of course to cause cell death, tumor cell death, to, in the end, in the tumor, in the patient, release and expose all the hallmarks of immunogenic cell death, which then can be perceived by the host and by the immune system. And then this immune system can mount an immune response towards resistant tumor cells or to dormant tumor stem cells. As this initial approach was very time-consuming, working with just one microscope. We lately developed a multi-parameter high-throughput station in which we are able to multiplex assays. This fully automatized and fully robotized station has the ability to screen large-scale compound libraries and also whole genome siRNA libraries, and it is equipped with 3 IXM XL machines from Molecular Devices, which are located here in our multiplex readout arena. It is equipped with a fully automatized compound handling and management system, which is on that side, and all this part here on that side is put in a sterile environment, and we've got a robotized arm which can move forward and backward from the sterile compound and cellular handling area towards the multiplex readout arena. With this station, we can conduct the primary screens using our automated microscope. So we conduct our image-based ICD fingerprinting using the automated microscopes, and we can also rearray all the compounds that are interesting for us in an automated fashion using our automated liquid handling system. And then this rearrayed compounds, so our hit compounds from the primary screen, are then used for validation assays using the automated fax machine, using the fluorescent reader to detect luciferase, and using the commercially available ELISA kits also on the plate reading system. And so this is how this lately generated station looks like. So here we've got our automated liquid handling system from Beckman. This is a Motoman robotized arm. I would like to show you a movie. So if somebody on the other hand can play this movie for me. So unfortunately, I couldn't see any movie. Anyway, I do just keep on talking. Okay, very good. So I just got informed that the movie played. I couldn't see it. I'm sorry for that. Okay, then we've got the multi-parameter high-throughput screening platform from the other side, which includes the automated fax machine that you can see here, and which also includes our 3 IXM machines. So this is the imaging devices that we're using to conduct our high-throughput screenings. And I would like to show you another movie, which I'm going to try right now. So if somebody on the other. Again, I couldn't see it, but okay, so I just heard that it should be done by now. Okay, so with this, I would like to thank the audience for listening. I would like to thank the organizers again for giving me the chance to present, and I would like to thank our funding organizations and our industrial partners for financing this this amazing platform and for financing our work here. And with this, I would be happy to take your questions, and I will pass over to
Thank you for that informative presentation, Dr. Kepp. Before I introduce our next speaker, Jane Hesley, I would like to mention to our audience that following the next presentation, there will be 2 surveys where you will be able to provide feedback on today's webinar. I would also like to remind our audience to submit questions through the Q&A box at any time. I would now like to welcome Jane Hesley and her presentation.
Sorry, give me just a moment. I'll make sure my slides are coming up. All right, today I'm going to present a high-content assay for detecting the several measurements of cell distress. In particular, I'll show you an example of an autophagy assay using high-content imaging. I just had a quick introduction to autophagy for people in the audience who might not understand why it's important in cancer. For one thing, the proliferation of tumor cells can activate autophagy, and increased autophagy in cells is a survival mechanism. Now, autophagy is a normal activity of cells for recycling used organelles and spent proteins and things like that. And cells in survival mode, though, can be resistant to chemotherapies. So a lot of cancer research is looking at using inhibitors of autophagy in conjunction with chemotherapeutic drugs to increase their efficacy. We showed an assay using cell lines that are similar to neurons or are neurons. I used immortalized cell lines PC12, which is from rat adrenal gland, and also induced pluripotent stem cell derived neurons from cellular dynamics. Why would you want to use imaging for cell health assays? Well, for one thing, if you want to study subcellular events, and in particular identify rare events in a heterogeneous population of cells, imaging is ideal for this. Imaging also allows you to characterize the mechanism of action of your cell treatments. For example, in one experiment, we started off doing autophagy, looking at autophagy, and realized we were leaving a lot of information behind. So we added a mitochondrial integrity assay, and we looked at the neurite morphology, and then we ended up also adding a lysosome, measurement of lysosomes inside the cells. Imaging gives you the flexibility to do live cell assays, and it can be done in time-lapse. Movies can be generated. You can see what's happening in real time. Time-lapse assays can be done in very quickly, only a few minutes, or for several days. And then, in addition, of course, you can do endpoint assays using the fluorophores. This is a diagram of the autophagy pathway. So autophagy can be inhibited or stimulated at different areas on the pathway. Autophagosomes form, sorry, I didn't really want to go all the way. So autophagosomes form where a membrane will form around the proteins that the cell needs to digest, and then this autophagosome will fuse with a lysosome, and the contents will then be digested and recycled for cell use. And in this way, the cell can get the needed amino acids for continued metabolism. Autophagy can be interrupted or stimulated at the beginning, or before at the area where the autophagosomes are fusing with the lysosomes. And later I will show you we did some experiments, and some of the compounds that I will show you we used for modifying autophagy were chloroquine and verapamil. So our one solution was to put 4 different assays into one well. We used a FITSI colored fluorophore to look at the autophagosomes, a dark red for lysosomes, and nuclei were marked with just a standard Hertz dye, and then the mitochondria with a mitotracker orange. If you're using cells such as neurons that can be difficult to segment, you can also use the transmitted light images, just a bright field image, to segment the neurons and make sure that you are correctly measuring the neurite outgrowths. In this assay, an assay of this kind, as you can see, could probably generate a lot of data, and you may or may not want to use it all. You're allowed, with our software, to report out only the information you're interested in from these experiments. You can record out the information on a field of view basis, or on a per well basis, or alternatively on a cell-by-cell basis. And this shows a few of the examples of things that we were looking at. For example, the total area of the lysosomes, or the average area of the nuclei that were there, so that we could check for toxicity. Also, I won't go in depth, but you can also look at co-localization. In this case, I will show you a little bit where we showed co-localization of mitochondria with lysosomes, or autophagosomes with lysosomes. In this experiment, we used molecular devices Image Express Micro for image acquisition, the MDC Store database for storing the images and the data. Then analysis was performed with Meta Express software, and the measurements from the image analysis were again stored in the database with the images. And then those data were Acuity Express software was used to do what we call hit selection, or you can do a lot of graphing, hierarchical clustering, things like that in Acuity Express. I'll show you a little bit of data from that also. The assay workflow was quite amenable to one scientist running this experiment. On day 1 or day 0, you plate the cells. If they're neurons, they were thawed and plated. If they're PC12 cells, they're lifted and plated. And I used, I think, exclusively 384 well plates in the data I'm going to show you here, which allows you to save, not to use fewer cells, of course, and smaller amounts of compound. After 2 to 5 days, 5 days for neurons so that they can mature, a drug was added. And in the experiments I'm showing you, I ran a dilution series so that we could look at a dose response curve of some specific drugs that we know should impact autophagy or mitochondrial health. Then the cells were drug treated for 24 hours and then stained for the different organelles that we were interested in measuring. The staining took anywhere, depending on if we did all 3 or 4 indicators or just one, anywhere from 30 to 60 minutes. Then the cells were imaged while alive on the Image Express Micro. And generally, I was using data from my 40x objective for generating my curves, and I took also 60x images. And then these were analyzed with Meta Express software, and then that data was sort of collected and further analyzed in Acuity Express. So the goal of the screen was simply to measure the toxic effect of the compounds by looking at live dead, just the dead cells based on the nuclear size and brightness, and then to identify the mitochondria in the TRITSI wavelength, and identify the lysosomes in the Psi5 wavelength, and then identify the autophagosomes in FITSI wavelength. This picture shows a mask of as you can see here, the autophagosomes are showing up in green, the nuclei are blue, and the mitochondria are orange. The mask that I'm showing is, in this case, showing the autophagosomes being found by the pink overlay, and the mitochondria being found by the sort of electric blue overlay. Then you have the choice of using pre-configured application modules in Meta Express software for finding some of the common processes you might want to use. I used some of these modules in a custom module where I could actually measure everything in one analysis this way. Here is an example of the images we used and the kind of reaction we would see with our different compound treatments. On the left, you can see the cyto ID from ENZO marking the autophagosomes. With higher doses of chloroquine, you get a lot of autophagosomes building up in the cells. And these are 60x images. The next one shows what an example of the lysosomes would look like with Lysotracker Deep Red from Life Technologies. And then Mitotracker Orange on the right-hand side with valinomycin, which will inhibit the staining of mitochondria at higher doses. The custom module that is available in MedExpress can make your analysis very fast because you're able to analyze all of these different vesicles or punctate objects at one time. And this is a picture showing the nuclei, the lysosomes, mitochondria, and autophagosomes. And then you build a module and one by one identify each of those structures you're interested in. In this case, I'm showing that I found the autophagosomes. On the left-hand side is the image of the autophagosomes, and on the right-hand side is the mask of what was found. Same thing for the lysosomes. Then in the Tritzy channel, finding the mitochondria, and even going a little bit further then, and this shows the autophagosomes and the lysosomes where they are overlapping. Simply, it's just a logical where the masks are overlapping. These are areas of co-localization. And same with mitochondria and autophagosomes. And the third one. So what this allows you to do is get the multiple outputs from every single well. In this case, I'm showing 3 different compounds that illustrate very well a dose-response curve. For example, verapamil showed the lysosomes increasing with increasing concentrations of the compound. The mitochondria drops, the mitochondria intensity or area would drop as the valinomycin dose increases, and the autophagosomes would increase with chloroquine. In addition, you can count the nuclei and see if you have toxicity. In this case, the black square is a control, an untreated control, and really the only one that seemed toxic was the valinomycin over higher doses. And those are the results from the PC12 cells. Then I moved on to doing experiments with the eye cell neurons, human neurons, and these are examples of what the images would look like with the neurons at 60x concentration. Exact same compounds, same fluorophores. But the challenge with neurons is that the structures that we're looking at can be located far from the cell body, and it can be hard to identify them and pair them with their cell. So in this case, I know it might be a little bit hard to see, but the mitochondria are orange and the autophagosomes are green, and they, in many cases, are located far from the cell bodies. So a way we can overcome this issue is to build a mask using the transmitted light image. At the same time as we're taking the fluorescent image, we'll take a bright field image, and we can build a mask from that using some of our modules such as neurite outgrowth. In this case, the mask is showing the whole area of the cell is in teal that was identified by the bright field image, and then the mitochondria are showing up in royal blue, and the autophagosomes in pink, the nuclei are yellow. And then even areas of co-localization, which there's few, and it's sort of hard to see. But I'll show you the mask, and then here's the bright field image that mask was generated from. And this allows you to also only identify objects that are fluorescent that are associated with a cell. If you have artifacts that are not associated with a cell, they would not be counted. And once the assay is run with the neurons, we can look at in Acuity Express, which is great for looking at batches of plates. These are 384-well plates, 6 in a batch. Or you can also easily see where your responses are by looking at a heat map of the plate. And you can drill down, we call this NDC Earth, you can drill down in Acuity Express and actually look at the cells themselves, the images of the cells and the masks that were generated. If you would want to screen the neurons in high volume, which is really the idea to do screening, you can speed up your image analysis with parallel processing. It is very intensive to analyze neurons with neurite outgrowth modules and with any neurite outgrowth analysis. And you can also use Acuity Express, as I showed, for batching plates. And Acuity Express will generate curves such as these that we got with the neurons, showing lysosomes, autophagosomes, and mitochondria toxicity. In the end, what you want to see is some kind of a result. Here is showing that we had fairly decent agreement between the PC12 cells and the neurons. Autophagy is very cell dependent. I've seen that you can get very different IC50s with different cell types and sometimes no response with certain cell types. In summary, I wanted to just say that I hope you can tell autophagy assays, even of neurons, are compatible with high content imaging, and that the user customizable image analysis setup provides you with the flexibility to measure a lot of different parameters at one time and only to output the information you're interested in. And once again, the aspects that I used from molecular devices. I would like to acknowledge my co-workers that worked on this with me. I had assistance from Oksana Sarenko, Steve Wiltgen, and Sindhu Galamudi was my really useful summer intern. And then Cellular Dynamics International for collaborating with us with their neurons. Now I will send the
Thank you for that enlightening presentation. At this time, I would like to invite our audience to fill out 2 short surveys. The first one asks, on a scale of 1, strongly disagree, to 5, strongly agree, I find the webinar content informative. Again, that question is on a scale of 1, strongly disagree, to 5, strongly agree, I find the webinar content informative. Thank you. Okay. Next question is, I'd like to request pricing or a demo on the ImageXpress Micro XLS system. While everyone's answering the survey questions, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Okay, and our first question is, this question will be for Jane. Can this be run as a fixed cell endpoint assay instead of live cell?
All right, yes, certainly. Oliver mentioned in his talk there is a marker in the membrane of the autophagosomes called LC3B. And there are antibodies against LC3B. You can build an assay with the LC3B, which works in fixed cells. My only advice would be careful with the permeabilization of the cell if you're doing a multi-plexed assay such as I did, because you can destroy the staining of your lysosomes or your mitochondria. Sorry, I'm trying to give it back for the next question.
Okay, and the next question is for Oliver. Oliver, how long does it take to do such a screen?
So actually the time limiting step is to, first of all, to generate the biosensor cell lines for the specific screen that you're doing. Once this biosensor cell line system is up and running, the actual screen, the primary screen, this is basically done within a week. Then you can dwell, of course, in all this amount of data for a certain time to identify hits. And then the validations, as we have been using and we are still using this enormous platform, can also be done in a very shortness of time. And then the real time limiting step is to do all the in vivo validations, because looking for this immune system related effect takes at least a month to see effects. Doing all this with a sufficient number of animals, of course, then can occupy a certain amount of time. But the initial screen, the image-based screen, this is done basically within a week.
Okay, our next question is for Jane. What can you do to really resolve individual mitochondria or autophagosomes?
Okay, well, first and more important is to optimize your staining. You need to have really bright staining against a dim background, and so that's really the best. You can get the best results from the best images to start with. And then some things you can do with imaging, of course, is to use a high magnification, use a high numerical aperture objective. I want to say a caveat with the high magnification is only use as high as you really do need, because it's important to get a significant number of cells in your field of view, or you'll have to take multiple fields of view to generate nice statistics. And okay, so to resolve the small objects, also in MetaExpress software, when you're acquiring, it is a wide field imager. The images I showed you were done with our wide field imager, but we have in the software, you can do digital confocal, so there's a software algorithm that will help you bring out dim or small objects in your image. And then once you have the images, there are image processing steps that you can take to further enhance your objects of interest.
Okay, our next question is for Oliver. Are you interested in other cell death pathways, necroptosis, others, RIPK activation?
Oh yes, of course. We are, unfortunately, there has not yet been a reliable image-based biosensor developed to detect the activation of RIP kinases. So necroptosis definitely is something very interesting for us, and we're eagerly looking for tools to run this in an economic high throughput fashion, not using massive amounts of antibodies.
Okay, let's see. How long do you acquire image? And this question is for Jane.
I'll answer that two ways, because I'm not quite sure what the question's getting at. Well, how long do we acquire images? Like, actually is, you know, a few, how long does it take? Say a second per well for multiple colors. And then if you're asking how long over time, my images, I didn't take mine as time-lapse, I took them as single points. I mean, I did take some time-lapse movies, but didn't analyze any of those. So the whole plate takes, if you did an entire 384 well plate, I don't know, 30 minutes or something, depending on if you had multiple colors and how long your exposure needs to be. The exposure is generally a few hundred no more than a few hundred milliseconds per color.
I believe we have time for one last question, and Jane, it will go to you. Do you use the NGF to differentiate the PC12 cell prior to the experiment?
Yes, in some of the experiments we did use growth factor to cause the PC12 cells to send out more outgrowths, but in a lot of them we did not. We did put them on a treated plate to encourage, adhesion, for them to adhere.
Unfortunately, we're out of time. I want to thank the audience for their interesting questions and participation in today's event. Thanks to all who submitted questions. We will respond in writing to those we did not get to today. Today's webcast will be available for on-demand viewing through June 2015. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.